Okay. Hello, everyone. Let's give it another minute and then we can get started as everybody joins. All right, I'm going to get started here. So welcome to all for this fifth webinar in this short course on drug repurposing. It's been a great series with a lot of information. We started way back in November of last year. We started, the first topic was on from the patent perspective, so patents in terms of drug repurposing. Then we had a second session talking about um, the university and industry perspectives on drug repurposing. We had a third talk, which was more regulatory focus from a regulatory expert. And then the previous talk, the most recent talk was regarding funding. We had two different nonprofits that were on and talking about their perspectives from the nonprofit side and the different funding opportunities that could be available. So the format of this particular webinar, which I'm excited about, so this is this is the finale of our short course, and it's a panel that we have here of experts from industry to provide different perspectives. And the format's going to be so I'm giving my remarks here. I'll then have each of the speakers give a brief presentation, including case studies. And then after that, we will have a Q&A time, Q&A discussion. So I'm going to, welcome any questions that the audience may have, and including questions that are specific to projects that you may have going on right now. Of course, keeping in mind to keep these non-confidential, but welcome those type of questions to keep it interesting and keep the panelists engaged. So I'll be monitoring those throughout the discussion, and so you can just put that in the chat box. So myself, for those of you who don't know me, my name is Raja Krishnan. I am a commercialization manager here at University of Kentucky. I manage a portfolio of technologies that are focused in the life sciences area. So it includes inventions that come out of the College of Pharmacy, the College of Medicine, and some from biomedical engineering as well. And so I've been at University of Kentucky for a little more than 2 years. Before that, I spent some time consulting for various startup companies. And before that, I spent more than 15 years in the pharmaceutical industry. Excuse me. And while in the pharma industry, I had the opportunity to work on, developing new products, and some of these products were repurposed drugs going down the regulatory 505(b)(2) pathway. So I do have some perspective on that as well. So that's a bit about me. Let's move into, we wanted to find out more about the audience and who's here today. So let's move into two different polls that we have here. So let's go ahead and put up this first poll. So this first poll is just to get an idea if, who in the audience is actually working on advancing a drug for a repurposed indication. So if you could go ahead and make your selection, then we will get an idea of who's out there. And whenever we think we have enough votes, I guess we can go ahead and display it. Okay, so I guess a fair amount have not done this before, so that's good for us to know. So yeah, let's go to the next question. We have one more poll question. Okay, I think we had a little technical difficulty there. Yeah, if we can get the second one up, yeah. All right, so we're having some technical issues. Anyways, we got a good sense of what where the audience is at, and hopefully at the other poll question was going to be about, yeah, here it is, it's up now. From what all of you out there have learned, maybe from previous webinars or others, things that you've attended recently, are you more willing to consider drug repurposing in the future? So we'll give a minute or two. Okay, well, that's good. That's good to know. So these things are of great value. So, well, we'll move right along then. I think we've got an idea, and so I'm gonna go ahead and introduce our first speaker, and I'm excited about this because Jayanti is an alum of University of Kentucky. So it's an honor to have an alum come to this panel and be one of the speakers. She's an alum, did her postdoc at University of Kentucky. But let me start with her introduction. And so she has more than 20 years of scientific and leadership experience in drug discovery, development, drug delivery. She's right now the founder and principal of JR Pharma Consulting, and that's based in the Bay Area in California. She has previously she had founded a team at ALS Therapy Development Institute. That was a nonprofit biotech based in Boston. She helped that organization grow to become the world's largest biotech solely focused on ALS research. She's got numerous scientific collaborations with academia as a PI and with industry as well. And a lot of what she had done does fall under the overall umbrella of drug repurposing. And what she did at ALS Therapy Development Institute, that team's efforts led to an IND for one compound, and there were many more in the pipeline. So, and they developed a lot of tools to effectively dose compounds. And so it was a great achievement there. She was also the finalist for the Academic Scientist of the Year Award by Informa Sciences in Boston for her outstanding contribution to pharmaceutical research. That was in 2004. So her initial interests were from the academic side was in controlled drug delivery systems, and that's what led her to UK. And she was a postdoc under the well-known and late Dr. Pat DeLuca in the College of Pharmacy. And thereafter, doing her postdoc, she was a senior scientist in different biotech startup firms. And she also has a lot of different patents. Finally, she holds a PhD in biochemistry from the University of Madras in Chennai, India. So I will hand it off to Jayanti. Welcome.
Thank you, Raja, for the nice introduction. Good morning, everyone. Excited to be in this panel discussion. I know this is the culmination of 5-part series of on drug repurposing. And very happy to know that though many of you are not currently pursuing drug repurposing, you are considering doing it in future. So maybe we can share some of our experience today, and maybe you can have a lot of takeaways at the end of the session. So let me start with, what are the lessons learned while I was pursuing drug repurposing way back in 2000 when the term was not so popular as it is today. Next slide, Jackie. So why repurpose drugs? I'm sure many of you are already aware of the importance of drug repurposing. The number one reason is drug repurposing keeps patients at the focus first, patients first. And mainly it helps in reducing the cost and the time to development. And who doesn't want that these days? So it's going to be the future path of drug development, especially for rare and ultra rare diseases, where the focus is not so much on these by major pharma, primarily because of lack of ROI. And by which I mean that, the patient population is very small. It's less than 200,000. So the amount of money that has been spent in drug development, as you all may be aware, it can take anywhere from 13 years, 10 to 13 years, and $1 to $3 billion. That's a lot of money to spend on diseases which don't have that much patient population. Clinical trials become an issue sometimes. So here we are. Drug repurposing is going to change the landscape of drug development in future. And the other reasons are that, there is a huge availability of data mining and data science these days, that you can see there are databases which are set up for FDA approved drugs with all the information that can be captured during the development, right from target identification, lead development, lead optimization, all the pharmacokinetics, in vitro, in vivo characteristics of the compound, and all the way to translational research and the clinical trials, and what all the way up to approval, and of course post-market surveillance on these compounds. But what is missing is, there are thousands of drugs that actually go through clinical trials every year, and most of them don't get approved. It's only 10% of compounds that enter phase 1 get approved. So that's a very small number there. And so all those compounds which have failed in clinical trials, now you have at least, some of them have gone through phase 1, they have good PK data, some of them have gone up to phase 2, and you have good safety data, and maybe in the efficacy endpoint or the target, or for some reason it failed in phase 2. So you have a lot of human use data from these compounds which have not been captured into proper databases, and they have not been shared. There is no open access to the public to get this information. So that's where the biggest chance for drug repurposing lies, because you have this huge database of compounds which are lying with pharmaceutical companies. So they are in a dead end if they are not shared. Trillions of dollars being lost right there. And because clinical trials are the most expensive part of drug development. And on the academic side, there are a lot of discoveries that are happening, but there are no incentives for them to share their data. And primarily the focus happens to be, unfortunately, on publications, and it ends right there. So you see this huge valley between academic research, very promising discoveries coming out of the academia, and the industry, which is pursuing ideas sometimes from the academia, sometimes on their own. But in between, there are so many compounds which are just lying dead, which could be revived and which could be taken to the clinic, and which could be repurposed for several indications. And as we are all aware, COVID is unfortunately, though it's a pandemic, and it unfortunately opened the eyes of many scientists, biomedical scientists around the world. And currently, there are 8,000 therapeutics which are being explored for COVID-19. Though we have the vaccines, they are still not enough to, save patients from hospitalization and death. So out of these 8,000 therapeutics, we found that 10% of them are repurposed drugs. Next slide, please. So as soon as the pandemic started taking hold, NIH opened up its COVID open data portal to openly and rapidly share COVID-19 data to researchers around the world, and which captured, all the data from assays, experiments, all the failed trials, all the positive studies, both from preclinical and clinical side. So they have data available for about 10,000 compounds, out of which 3,000 are approved drugs. So there you can see the importance of drug repurposing, which was taken as a lead by NIH. And as you're aware, even for the vaccine development, all this could happen only because of collaborations and partnerships between university and industry. It's a key thing in drug repurposing, because you're going to start with a compound which is approved, which has probably gone through several hoops from academia to industry, even within the industry, from small startups to medium, mid-sized pharma to big pharma. So the collaboration is the key, and I think that's going to drive drug repurposing. So for COVID, the most recent data on the drug bank shows that there are 62 drugs which are in clinical trials, addressing 84 different drug targets. Total number of clinical trials, an astounding 3,308 clinical trials ongoing, and the publications are 54. Early on, if you see in drug development, the publications and patents would be high on the list. You see now here, people are open to testing something fast and something that can reduce cost. Next slide, please. So this is just, this is shared on LinkedIn by someone. These are all the potential targets that have been pursued for COVID-19 repurposing. Here's the virus, and you can see that numerous pathway analysis and network assay or something they call. They have a lot huge pathway networks where they found, the potential for targeting COVID, the spike protein, and many other parts of the genome is very, very high. Next slide, please. So just for the cell culture studies, if you see that the approved drugs that have been studied for COVID, it's an astounding list. I just picked a few of them here. And what you can see is, many of the compounds come from, they're antiparasitic, anti-inflammatory. They come from different disease indications, anti-pancreatitis, compounds which are for obesity, cardiac compounds. You also see a lot of immunosuppressive compounds which have been in clinical trial. So the opportunities are immense. Once the understanding of the pathways are reasonably good, then it goes, it reduces all your early development time if you take a repurposed compound. Next slide, please. Next slide. Yeah. So currently we have 3 different options for COVID as repurposed drugs. You may be aware of Gilead's remdesivir, which has been used for as more for managing or preventing hospitalization in the early stage of the COVID treatment. There is also 2 antibody cocktails, one by Regeneron, the casirivimab and imdevimab, and the other one is from Eli Lilly. There are also several other compounds being pursued as repurposed drugs for COVID. All this to expedite, translating drugs to patients as quickly as possible. Next slide, please. So as Raja mentioned, my experience with drug repurposing started with ALS Therapy Development Institute, which was founded by Jamie Haywood when his brother was diagnosed with ALS at a very young age of 29. He was at Scripps Institute in the Office of Technology and Licensing, coincidentally at that time. So he just quit his job right then, drove all the way across the country to Boston to start this institute to see if there is any opportunity at all to find something which can make him live. The reason from diagnosis to death for ALS at that time was 18 months to 3 years, very, very rapidly progressing, devastating disease. He found out, so I joined at the outset when they were trying to form the drug discovery program there. So what we know about ALS, we said, how are we going to go about this? We don't have much time. 3 years is too short a time to develop any drug for any indication period. And we have a patient whom we know who's going to be progressing day by day. So the first thing we did was a need assessment as to try to understand what the disease is about. Not many people had heard about it. So we contacted a lot of neurologists around the country in leading hospitals. And we also tried to, I mean, of course, code the literature to get as much information as we could. So what we found was astonishing. It affects only fewer than 30,000 adults at any time in the US. And there was only one drug, Riluzole, which is a Novartis compound, which was out there. And it does not cure the disease. It was approved, which gave only 3 months of improvement in quality of life. So no guarantee, the progression was not stopped. It just was marginally effective in giving better treatment to patients. So there it was nothing out there, and patients were suffering. There were numerous clinical trials that are happening. It was astonishing, the numbers. Several hundreds of clinical trials are going on at that time, both in the US and Europe, and some in Japan and Italy. And most of them were actually targeting symptomatic improvements. None of them were really trying to address, the fundamental cause of the disease. So from our standpoint, we were a nonprofit biotech that we started out in 2000. The stakeholders were patients and caregivers. So we had limited resources, and we had to work with the patients. We had to work within the constraints to see if we can come up with something that could make a difference. So we came up with an innovative drug discovery platform as an alternative to traditional drug development. And we looked around to see, okay, now that we have the program, how are we going to test the compounds? Where are we going to test them? And there were few animal models that were in development. And we said, okay, at least that's a good start. But there was no gold standard. There was no single model that was kind of, the best model out there. Because the clinical trials are clearly failing at that time, most of them. And the disease mechanisms were not fully understood. Several pathways are implicated, from excitotoxicity to oxidative stress to immune modulation or immune dysregulation. A lot of pathways have been, thought upon, but none of them were proven at the time. And as I said before, major pharma were not interested in drug development for ALS because of lack of ROI. Very disappointing at the time. And patients had a very short time to live from diagnosis to death. So the urgency was felt by our team, and we said we have to do something different. Can I have the next slide, please? So I think the previous one. Oh, okay. So we said, okay, we have to go through an accelerated drug discovery strategy. Yeah, the next one. Thank you. So what we did was we had to come up with a new process. And at that time, when we were starting in 2000, it was not termed as drug repurposing. It was not so popular. Drug repositioning, nothing was, a fancy way to pursue drug development. So we said that we had limited resources and we had limited time. So we said we ought to do this through collaboration. And we said, we're going to go out to every academia and industry which has got anything to do with neurodegeneration or working on musculoskeletal diseases. And we would approach them and see if they could help us with taking this program forward. Time was the essence, and the road from discovery to patient had to be really short so we can actually turn something meaningful to patients. And we also decided that we understood very little about the disease from the patient standpoint. So we engaged patients and the clinicians throughout the discovery process, right from the outset. We in fact had patients come into our lab and we would see them progress. And I can tell you, it has been so disheartening to see, I would have shaken hands with somebody just like you and me. And through the 9 years that I spent there, we had a lot of people who moved from that stage to wheelchair, and many of them passed away. So we could see the urgency of drug development that was needed to help these patients, at least, if not cure, at least something to slow the progression. So the approach we took at that point is, we said the end in mind is bring a human context to therapeutic discovery. And we have wanted to minimize the early discovery timeline, which is what drug repurposing does. And for that, we approached the industry mostly, where they already had compounds on the shelf or in the clinic that have been thoroughly characterized in terms of target, in terms of the lead, and also in terms of pharmacokinetic data, in terms of safety and efficacy data, primarily for safety reasons. So we could directly put the compounds into the animals and test them. So we focus, since we had that buy-in from some of the collaborators, we said we will focus on making sure that the disease model that we are testing is robust, because that makes all the difference between a compound that can be successful in the clinic or something that's going to fail. So next slide, please. So what we did was we scouted around to make sure that we had the best model in our hands. We characterized the model to make sure that we'll get reproducible results. And that's when we learned that many of the compounds which were in clinical trials at the time, most of them failed. But the reason is, when we tested those compounds with the exact study designs that they used in our the lab, but we used systematic study design. We had enough number of animals and a very sound study program. They were not reproducible. We tested again and again and again when we had lots of failures, and we learned that the reason is, when we went back, we found that the model itself was not thoroughly characterized. There was not enough information out there, and people are trying to do in uncontrolled studies, and either not gender controlled or the numbers were small, but they were taken to the clinic because of the urgency of need. So we decided to have a rigorous program with standards established right from target identification all the way to preclinical efficacy. We had sound study designs and protocols. We also established LIMS to make sure that all our data is not only just loaded for internal sharing, but also for sharing with our partners. And we screened more than 100 compounds, primarily small molecules, but few other therapeutic modalities. And many of them were failures. Many of them were actually lessons learned, I would say, because the model is a very rigorous model. It's it does replicate the human disease, but the number of copies of genes is very high, about 100 times higher than what it is in humans. So because of that, the chances of success was very limited. And we couldn't do otherwise, because this model, the mouse lives anywhere from anywhere from 120 to 140 days, while all other models, the low copy mice, they would live up to 18 months or even longer. And it was very hard to move anything faster with that kind of a model. Next slide, please. So the favorable outcomes that came out of our research is, first, we came up with an innovation. In a way, a lot of innovation happened, where we first modified the model that it became the gold standard for testing ALS therapeutics. A lot of technology development, because we had to, the understanding was that the disease is affecting the brain and spinal cord. So you got to make sure that the compounds that you administer get to the brain. So we pioneered in developing a technology, intrathecal delivery to the brain for 30 days in mice. The size of the mice, and to give something chronically for that long was not out there in the industry at that time. So we were able to develop that thanks to all our in vivo technicians, tireless efforts from them. And a lot of other tools, formulation tools were identified, formulation strategies were discovered. And we also established, a large scale screening, in vivo screening platform, which was again not known in academic or nonprofit settings. We would screen at least, 4 to 8 compounds per month, which would be, any of them would be either PK studies or acute safety or proof of concept efficacy testing. We established numerous collaborations and partnerships, at least 45 to 50 when I was with them. And really good relationship that we had with all our partners. We had partnerships both in the US and globally. A lot of data sharing. We emphasized on data sharing and openness with our partners. So ultimately, the outcome was a collaborative drug discovery platform with high quality data that you can rely on, data that is true. And of course, the entire program was focused on patients. Next slide, please. So those are what we learned and what we had. But going through the program, we found that there can be numerous ideas that can actually help drug repurposing in the current times. One is, the formation of a consortium, which could be either disease specific or it could be technology specific. If you're a company who's working on drug delivery, then you can actually form a consortium of drug delivery for CNS delivery, or it could be for subcutaneous, it could be infusion, something which is applicable in patients. It could be a medical device. But as long as there is a consortium which consists of people both from the academia and the industry, so the R&D leads to something meaningful for patients. I think putting patients first should be the focus in future for drug repurposing. The second one is to have an open access centralized data bank of both FDA approved compounds as well as failed drugs, which will include all the research data that has been generated with taxpayers' dollars through several years. Very, very useful tool that can be developed. There are data banks now currently available at NIH as well as distributed among several universities or some of the industries, but they are not centralized. The only one that we have is the NIH NCATS database and the drug bank, but they're not inclusive of all the failed drugs, where there's a lot of opportunity. And there comes, the application of predictive artificial intelligence and machine learning, because a lot of information can be gleaned from this type of data to advance molecules faster and help with lead optimization. And last but not the least, I feel drug repurposing is also eco-friendly because you're maximizing the resource sharing, you're minimizing repetition, you're minimizing wastage of resources, where supplies, equipment, assay development is repeated across R&D, both in academia and industry. So ultimately, reduce cost to the clinic and help the planet in a way. Next slide, please. So I think I didn't mention this before. So currently I'm having my own consulting firm, but I'm also an advisor to SPARC, which is Stanford's Translational Research Program. This is one classic example of how an open relationship between academia and industry can lead to really sound science and treatment for patients. So this program was established in 2006 to advance academic biomedical discoveries into potential treatments for patients. The directors there are Daria Mockley-Rosen and Kevin Grimes, both of the Department of Chemical Biology, and Peter Santamaria. And the partnership is a unique partnership between SPARC program is a unique partnership between the university and the industry experts. So all the technological advances that are happening in the academic side at Stanford is translated to the industry or to a startup mode by bridging the gap between the two. The Spark program just lies right in between, where they get the help of industry advisors who are experts from several different areas of the industry. They come every week. We meet at Stanford, and both postdocs are the Spark program participants are called Spark scholars. They go through a rigorous drug development graduate level course, and they are also open to a lot of drug development seminars and webinars and workshops, where they learn some of the tricks, as well as some of the important things to consider in drug development, especially going from the academia. And the outcome of this is not only just research, but also new diagnostics, new biomarkers that have come out through this research. Next slide, please. So as I said, it's the unique partnership between industry and the university. And the way they connect the industry with the academic researchers is through active collaboration, inviting industry speakers to come and give presentations at the Spark meetings. I've attended several talks from Genentech, Amgen, from several companies in the Bay Area, primarily because it's local, it's easier, but now with the pandemic, everything is online. So we have industry experts coming from around the country. The program is open to all professors, postdocs, clinicians, and grad students. And as I said, the advisors help with, a lot of things go with, product development. Sometimes, the advice given is very, very small. The details are so small, but they're so vital in drug development. And we have a lot of clinicians who come in to inform about, is this practical? Is it translatable to the clinic? We may have a great idea. The product, the drug could be having a lot of potential, but delivery could be an issue. So the clinicians come in to give us some valuable input to see if this can be translated at all. There was one talk from Genentech which talked about target product profile, which was not thought of in the academia. Academia, you go with, can I see if the fundamental mechanism is sound? Then you go to the lead, and then you go to optimization. But as I said, start with the end in mind, where major biopharma, they first think of how is the patient going to use it? How can we deliver it? Can this be dosed? Then they go backward. So if they are looking at an oral compound, they'll say, so I'm going to have a tablet, it's an oral delivery. And then this is the patient's thing, the time frame for admission, for treatment is anywhere from, few days to weeks or months, or is it going to be a chronic treatment? So you write your product profile and you go backward. And I think Kristen will be talking a lot about it with a good example. So again, the program is based on 5 principles. Multiple opinions are okay. No need to reach a consensus. We all discuss. Open exchange, and there is no hierarchy. Everyone contributes equally. It's on the university campus, so it gives that intellectual atmosphere. And it's a 2-year program they have with $50,000 per person. The money is given by Spark, and additional money comes from investors who are from the industry. Next slide, please. So what are the success stories out of Spark? I can give you 3 examples of repurposed drugs which came out of Spark partnership. And all these 3 people were part of Stanford faculty or postdoctoral research group. And they have started their own company. And some of them are very successful. They have huge potential. And so for the first one is Globavir, which is started by Dr. Shri Tainav. Her team worked on a combination of 2 FDA-approved anti-cancer drugs for hepatitis C, dengue, West Nile, and Ebola. So antiviral applications of 2 anti-cancer drugs. Then we had Mirad Shamlu. He founded Curacen, where he worked on the activation of beta-adrenergic pathway compounds. He knew that this can have a significant effect on the cognitive function of Alzheimer's patients, but just pursuing compounds directly addressing this can lead to heart failures. So Spark helped him in tweaking some of his development needs and found a small molecule candidate, which was CNS penetrable and was not only used for Alzheimer's, now it's being tested for Parkinson's also. The third one is Eiger Biopharmaceuticals, Dr. Jeffrey Glenn's team. They identified a compound, which was an anti-allergic compound, antihistamine, which has now been pursued for hepatitis C. Next slide, please. Okay, so that's the short story. And I just wanted to share some of the resources that can be used for drug repurposing. These are out there currently, open access. A consortium to combine all these would be great. So the first one is at Harvard Drug Repurposing Hub at Broad Institute. They also have a connectivity map, a database of all the, it's a where you can subject cells to perturbations, and they find out what happens with the disease or the genes or the treatment that you have given. And they've captured every single data point with these. And this has helped develop hypotheses for several indications through this map. And I think it's one of the classic reference tool now for drug repurposing. And at NIH, you have the NCATS pharmaceutical collection. Again, it's publicly available. You have a lot of information on the compounds, the primarily small molecules, but which have been approved not just in the US, but also Canada, Japan, and Europe. The next one is from San Diego University called Caliber, and the drug repurposing hub is called Reframe. And they have a collection of about 14,000 compounds. And really powerful tool. The next one is at UCSF in San Francisco and in Dr. Butte's lab. They do a lot of artificial intelligence and machine learning, early part of drug discovery, I would say, for target and lead identification, computational drug discovery. They are primarily focused on oncology target right now, but I'm sure they are looking at other diseases also. And the last one is called a data descriptor. This is a very recent one, again coming out of Harvard Medical School. This is more for finding alternative indications, but what they have is basically they have both kind of a failed compounds as well as FDA approved compounds. It's still in development, but certainly a good place to go to. So I think that's the last slide, Jackie. Yeah.
Okay.
So, yeah.
Thanks
for listening. Yeah. Okay. So now I think you can go on.
Yeah.
Thanks, everyone.
Yes. Thank you. Okay. And thanks for those that insightful sharing of your experience. And I wanted to just emphasize really quick the idea that was mentioned about thinking about all these things like the business related justification kind of earlier in the process and way up front. I think that is especially true when trying to move a repurposed drug asset, thinking about what lies ahead in terms of the business side of things, the intellectual property side, the regulatory side, really encouraging the thinking of that to occur as soon as you identify a novel target and start the process of trying to screen, let's say, FDA approved drugs. And we've talked about that in previous seminars as well, and just emphasizing that thought. So I will go on to the next speaker, and let's introduce Douglas Balogh. And he is a director in Guidehouse's Life Sciences Department of Disputes, Regulatory Compliance, and Investigations practice, is focusing on FDA regulatory matters. He's got more than 31 years of industry experience in drug product development and regulatory affairs. Before Guidehouse, Douglas extensive experience at Eli Lilly and Company, and it included his positions were in scientific and managerial leadership positions in the R&D part of the organization. At Guidehouse, his strong technical background is a cornerstone of his service to clients as a pharmaceutical regulatory affairs specialist. He's assisted clients in development of regulatory strategies, CMC strategies, and achieving successful INDs, also obtaining orphan drug designations and submitting 505(b)(2) applications. He's guiding clients through successful meetings with the FDA. So now we will welcome Douglas and to give his presentation. And one more thing, just to encourage again and a reminder, we welcome any questions. So please go ahead. You guys can put that in the chat box. So Doug, you're up. Thanks.
Okay. Thank you, Roger. So what I'm going to do is take 505(b)(2) projects where Jayanthi left off, because my background is development of candidates once they've been identified. And like Roger said, I was at Eli Lilly for 27 years working in product development. I've got experience in small molecules and biologics during that time. Left Lilly end of 2008 and have been consulting ever since, mostly in CMC and in regulatory affairs area. Most of my clients have already identified their lead molecule and are wanting advance advice on how to support their product, getting it into the clinic and getting through FDA's criteria. So they want to understand what FDA wants to know. And not surprisingly, they want to know what is the minimum they have to do to satisfy FDA in order to advance their product. So in this capacity, I've worked on a wide range of projects, including small molecules, biologics, gene therapy, cell therapy. A number of these have been 505(b)(2)s. So if we go to the next slide. So the 505(b)(2) pathway, you can be very creative with that. And it's repurposing sometimes in people's minds sounds like, well, I'm just taking something and I'm trying to make it fit something else. And really, my clients are looking at the 505 with new indications. They're looking at new delivery methods and they're trying to reduce side effects. I've got a list below of a half a dozen of them that I've worked with recently, looking for a new indication for an old drug. And I've got a case history on that I'll share. I've got another client working at subcutaneous sustained release formulation that's going to benefit convenience for the patient and importantly, attempt to reduce side effects. I've got another client that is using red blood cells as a delivery device they pack it with a drug and administer it back to the patient, and the drug pays out over a prolonged period of time. I have another client that's advancing a single diastereomer of a previously approved racemic mixture. It turns out that many of the side effects can be attributed to the wrong diastereomer. And so they help to use that to target a very specific new indication for the single diastereomer. I've got another client that's developing a smart capsule. They pack this capsule with a drug. The capsule is actually an electronic device, and it implants itself in the GI system. And based on input from an external sensor, it can release the drug on an as-needed basis. It's integrated with a smartphone so that the release of the drug is recorded, fed back to the user and ultimately the care provider. And then I've got another client that's developing a nasal delivery system for a previously approved oral drug. And it will provide kind of instant relief to the patient. And again, this nasal form has not been approved before, and they feel they can advance that and get a niche in the market. So let's move to the next slide. So to kind of summarize, finding a new drug, a new use for an old drug, sounds like maybe half the work is done, but there are still very big challenges that await. that need to be thought through and overcome during the development phase. And another, finding a way to reduce side effects certainly is a tremendous benefit to the patients, but to get there, there are a lot of questions that must be answered in development and constraints that must be considered. And Roger talked about, thinking through the end in mind. And Jayanthi mentioned this as well with the target profile. Thinking about how will this drug fit in the market really will guide what you're going to do during the development phase. So let's talk through 2 case studies on the next slide. So this is one that I got involved with several years ago. The innovator is a professor at University of Cincinnati, and he is his specialty is cardiovascular diseases, and he's a physician. And patients that have acute decompensated heart failure have a very poor prognosis. There are some drugs out there that are used. Usually they're ionotropes, but unfortunately, they're used and they help short term, but paradoxically, these medications have been shown to increase long-term mortality because they stimulate the heart. The heart's not in very healthy condition at that point, and it actually damages the tissues of the heart, even though it stimulates the heart and gets the patient through the short-term acute crisis. What he found was through his research that this transient receptor potential villonoid 2 channel can upregulate the systolic function of the heart. And he screened a number of compounds that could possibly treat that, conducted some research, and found that probenecid, a very old drug, approved a long time ago for the treatment of gout, stimulates that receptor. So then he set up a series of studies in the laboratory and found that indeed this receptor does regulate cardiac output, and moreover, using mice that have been compromised to stimulate heart failure, had their cardiac function return to normal levels with the administration of probenecid. So then ultimately the hypothesis was that this treatment could be used to treat acute decompensated heart failure, potentially minimizing or eliminating the long-term mortality. Next slide. So at this point, I was brought into the project, and he was seeking information on how does he get this into phase 1 clinical trials, and what would be the best regulatory pathway. And we selected the 505(b)(2) pathway because the drug is already on the market. There's a great deal of safety information about the drug out there. He wasn't really interested in the efficacy information because it's used to treat gout, but he wanted to rely on the safety and not have to repeat all of the toxicity studies. So we selected that pathway. He would need to treat heart failure an IV form of probenecid, because it's currently a tablet. So what we did was decide to meet with FDA, have a pre-IND meeting, and lay out our plans for development, get FDA's feedback. And at that meeting, FDA raised no major gaps that needed to be addressed prior to initiating phase 1, except for one. And the FDA agreed full animal toxicity studies would not be needed, but FDA requested local toxicity studies in animals be conducted. And that's not uncommon whenever you change the mode of delivery for a drug. But fortunately, after that meeting, we went back and the researcher found a paper in the literature that where probenecid had been given intravenously to humans one of these old drugs has been studied a lot, wealth of information and literature. And we were able to look at that paper. It was a well-done research paper. And from there, extrapolate how his planned dose schedule would map up with the dose plan that was in that paper. We wrote that up, requested a second meeting with FDA. They considered the paper and agreed that it was not necessary to conduct that animal study, and that in fact the investigator would be now allowed to, after the IND was submitted, begin phase 1 clinical trials. One point I want to mention, and Raja mentioned as well, the university's legal department had developed a patent strategy, and that strategy relied mainly on the new use and on the IV administration. Next slide. So where's the project now? Okay, well, after the successful pre-IND meeting, a venture capital company became very interested in this asset and in fact acquired it from University of Cincinnati. But after a year to 18 months, they wanted some additional patent coverage to make it a economically viable project for them. And they were not successful in implementing their additional patent coverage strategies. I don't know what those strategies were exactly, but they didn't feel like the use patent was sufficient to give them the return on investment they wanted. So ultimately, they spun the asset off back to the professor at the university. And he's got other interested investors and does plan to advance the project once he gets additional investment money coming in. So the key takeaways from this one are repurposing greatly increases the chances for clinical success because the safety of the drug is already known. You should definitely rely on scientifically sound literature studies where appropriate to reduce development costs. Gain alignment on your development plans with FDA, and a successful meeting with FDA can help obtain venture capital or maybe even spin the asset off. And finally, prepare a sound patent protection plan. 505(b)(2) with an existing molecule gives you 3 years of market exclusivity. For many investors, that may not be sufficient to give them the return on the investment they want. Let's do the next case study. Next slide. Okay, so there we go. So on this one, this project, instead of being developed by a university professor, was developed by a team of polymer scientists. They've had some drug experience, but they had a lot of experience with polymers. And and I'll get into a bit of the details here in a bit, identified a medicine used to treat serious neurological disorders and felt that using their polymer technologies could develop a sustained release form of this drug. And by having sustained release, they can reduce some of the side effects that are caused by the peaks and valleys of taking an oral medication. So they wanted to develop the subcutaneous form of this drug, eliminate the GI side effect systems that come from the peaks and valleys, and pursue a subcutaneous approach for delivering the drug. So what they did was engineer a custom length polymer and then chemically link that to the drug. And by controlling the length of the polymer, they could control the release of the drug and release it over an extended period of time. And thereby, because it is administered subcutaneously, they could reduce and potentially completely eliminate the GI side effects of the drug. Next slide. So they needed regulatory support they came to me, and we quickly identified the 505(b)(2) pathway because there's a complete body of information about the safety and efficacy of the drug that are already in existence. And so and they also had their own patent strategy worked out, which mainly surrounded not the efficacy of the drug, but rather the construct of the polymer and linking the drug to the polymer. So that is a very sound patent strategy that would be very unique to them. So while the drug had been studied in animals and humans, the conjugate with the polymer, of course, had not been. So the client proposed a set of animal studies that would need to be done. Instead of the full 2 species of animals that's normally done, the client proposed, because of the body of information that's out there, that they could only study 1 species and save development cost and time. And there was a good literature rationale for that. So they proposed that to FDA. They also proposed that, of course, local tolerability studies would need to be conducted. Again, when you change the mode of administration, you would need to do local tolerability studies. And they also proposed to FDA that since the final polymer could deliver the drug at the same average blood levels as the reference drug, that they could rely on FDA's previous finding of efficacy. They supported that argument with data drawn from the product label and a number of research publications where the pharmacokinetic profile of the drug had been studied. And based on their animal studies, they were able to calculate that their product would be available to the patient in the same amount as the oral drug over a 24-hour period. Then with that information, we met with FDA. Next slide. So at the pre-IND meeting, FDA agreed with most of their ideas. The FDA thought the 1 tox species would be acceptable. Local tolerability studies would be required, of course, but FDA agreed to 1 species. What FDA disagreed with was the area under curve matching plan for the bridge to efficacy. FDA wanted bioequivalence to the reference drug. And FDA's rationale was that the exposure response relationship, in other words, if you give an oral drug, there's a peak spike, there's then a valley where the blood levels are very low, and then patient takes another dose and that cycle repeats. FDA felt that a continuous payout could not bridge to efficacy to a kind of a sine wave pharmacokinetic profile. And there wasn't enough data in the literature to support that that would be a valid bridge. So that now fell back on the client that they would now need to do bioequivalence studies, which would be highly unlikely given the to show bioequivalence given the way the drugs are administered. So therefore, they would need to be required to conduct full phase 3 clinical trials. Well, the project at this point is seeking funding to support further development. However, there's a small market size, and the investors are looking now to find additional funding to enable them to conduct those clinical studies. So again, this goes back to a point that that Raja had made, in that you need to think about the end in mind, how your product will fit into the market. And these clients felt like it was worth taking the chance with FDA about their efficacy bridging study before they spent a tremendous amount of money in development. Unfortunately, it didn't work out in their case. So the key takeaways: alternative modes of delivery should definitely be considered as a way to mitigate side effects. Research and rely on scientifically sound literature studies where appropriate to reduce development costs. Prepare a sound patent protection plan. In this case, they had a very good one with the polymer. And I think very importantly here, gain alignment on your development plans with FDA before investing in expensive toxicology studies. And I think that really is the key takeaway on this one, which this client did. So I think that is my last slide, and I will turn it back to you, Roger.
All right, thank you, Doug. That was really good, sharing those experiences, especially those cases involving the challenges faced by these different technologies when going the repurpose route. I think that really gives a sense of some of the different types of challenges that you could face. And also highlighting that the importance of patent strategy. It is highly important, and furthermore, to our researchers at UK, and I encourage other researchers at other universities as well, is when you get to a point where you have a novel target identified, and you're at the point where you're trying to maybe screen a library of drugs, let's say FDA approved drugs, that you also contact our office, right? Those UK researchers contact us at OTC and let us know what your plans are, because we could help you there doing a patent landscape analysis, and also confirming that whether the drugs that you guys that are being screened, although they're FDA approved, how much patent life do they have, right? So that's another key thing. Is it short, or is it going to be a longer patent life, or hopefully the ones that are have more business potentials are the ones that are off patent, right, or very close to being off patent. So these type of considerations we can definitely help you navigate. So again, thinking about those things up front. But without further ado, I'll introduce our next speaker, Kristen Gullo. She's the Vice President of Development and Regulatory Affairs at US WorldMeds, which is based right here in Kentucky. The company develops, licenses, and brings to market unique healthcare products that are designed to improve the lives of patients with challenging conditions and unmet medical needs. Kristen provides strategic leadership for all elements of pharmaceutical development, including CMC, preclinical, clinical, which would be phase 1 through phase 4, and other human factors related to studies, regulatory interaction, and data dissemination. She interacts with commercial teams for alignment of clinical development programs to fit market objectives, and is knowledgeable in GCP, GLP, and CGMPs. So I welcome Kristen and look forward to the presentation. Thank you.
Thanks, Raja. Well, I'm going to follow my other very qualified panelists and really try to highlight a case study in the area of drug repurposing. I'm hoping you'll take away from my presentation a few things and really pull through some of the concepts you've already heard from the other panelists. I also want to highlight this specific example because it is also a really nice example of academic industry collaboration. And there's also a philanthropy component. So I think you've heard a little bit about each of those so far. So we can go to, well, actually, just to introduce the topic, the program I'm going to review here is DFMO, also referred to as eflornithine, for the treatment of high-risk neuroblastoma. And to just, I guess, introduce the format of my presentation, I'm going to kind of start with a general question. I think it's a general question you should ask yourself when you're approaching a drug repurposing program or considering one. And then I'll take you through how we answered this question, in my team to decide to take the program on and then actually move it forward. So the first question is, what is the history of the molecule? In the case of DFMO or eflornithine, start, of course, with the mechanism of action. So DFMO is an ornithine decarboxylase inhibitor. The role of ornithine decarboxylase is it is a rate-limiting enzyme in polyamine synthesis. The eflornithine molecule has had 2 prior approved uses in the US. It was approved as Ornadil by Sanofi for African sleeping sickness. They actually still currently manufacture that product, but it is exclusively distributed by the World Health Organization. But the thing to note about the use of DFMO and Ornadil is that this prior approval established safety of high doses of the medication in an intravenous formulation, as well as they had generated safety data on both adult and pediatric use, which will be important as we describe the condition next. But the other approved use is by a subsidiary of Allergan, and is called Vaniqua, and it's for unwanted facial hair in women, and it is marketed as a topical cream. So we can move to the next slide. So, the next question is, thinking specifically about this mechanism of action, does it have relevance in another area? And in the example of DFMO, there has been a lot of interest about the mechanism of action as it relates to oncology. So polyamines are known to be essential to cell growth in a variety of cancers. Probably over 2 decades ago, this molecule got a lot of attention from the National Cancer Institute. And we'll cover in a second that they actually invested heavily in producing GLP tox studies to enable INDs to be opened in oncology and pediatric oncology. So there's a lot of interest, but to date, there still remains no approved uses of DFMO for any oncology indication. So, in our specific situation, we focus on high-risk neuroblastoma. So I'll just briefly introduce that disease. So there's there's 800 cases of neuroblastoma diagnosed every year in the US, but about half of those are considered high risk. The thing to really know about high-risk neuroblastoma is that it is primarily diagnosed in children under the age of 6, although it can cover the full childhood age range. And, it's a very devastating disease. In particular, the high-risk category has a very poor long-term survival of, the literature estimates around 50%. So as with a lot of cancers, a lot of children are able to achieve initial remission with the therapy that has been developed over the last, I'd say, 2 decades. And so, the proportion of patients that do get to an initial remission is pretty favorable. However, the risk of relapse remains extremely high once treatment ends. And the long-term survival of 50% really reflects the devastating effects of patients that do relapse, because relapse disease is much more aggressive and much more difficult to treat. Relapse is linked to minimal residual disease. And, the hypothesis is that minimal residual disease includes some level of dormant cancer stem cells. And the research in particular, the original animal and in vitro research with DFMO in neuroblastoma does support that polyamine levels are suppressed by DFMO. And then there's also been some really exciting research into the specific effects of DFMO on the neuroblastoma cancer stem cells. And it does support that it does support DFMO appears to selectively target those cancer stem cells and actually can result in cell death. We can move to the next slide. So as you've heard from Jayanthi and from Doug previously, we really do have to think with the end in mind when we approach drug repurposing projects. And you've already heard about the target product profile. So this is, a term used in drug development to really just say, when you get to the end, what do you have to be successful? And just very generally speaking, for this DFMO project, you heard that previously the product was developed as an IV formulation and then as a topical cream. Of course, we need good bioavailability, but we're really talking about the specific focus on preventing relapse over a 2-year period. So we need a formulation that can be dosed in an outpatient setting. And there was prior work, which I'll discuss in a little bit, about the oral dosing of DFMO, which does establish sufficient bioavailability so we know oral dosing is feasible. And then our selected formulation or options for a formulation that we considered was a tablet that can either be easily crushed or dissolved for administration to pediatric patients, or an oral solution. We needed a formulation that would enable dosing across a large age range of patients. So a lot of oncology products are dosed by surface area. So we needed something, to be able to adjust the dose by surface area that would cover a very large age range, basically going from, infants to young adults. Most diagnoses occur under the age of 6, but rarely diagnoses do occur in adolescence or even early adulthood. We also knew that we needed something that was really well tolerated. The difference in considering a medication for maintenance of disease is different than the sort of risk tolerance or the safety profile tolerance that you can have for acute use, especially in oncology. I mean, we know that a lot of oncology products are unfortunately highly toxic, but the benefits outweigh the risks, especially in an acute setting. But it's not reasonable to think that you could accept, a very toxic safety profile for chronic dosing in children who are now, in remission. And the goal of the therapy is to keep them in remission and have them be able to participate in a normal childhood. And then, of course, on the efficacy side, we really had to aim for something that was meaningful to patients. So we set our goal at a minimum of 10% improvement over standard of care in relapse prevention. Next slide. So the next question, after you have your end in mind, is what do you have to work with? So the B2 mechanism does allow you to leverage existing data in the public domain, or that you can get right of reference to. So the question is, can the historical work both be obtained and successfully leveraged? So in this case, we were able to determine through literature review that there was an extensive non-clinical toxicology and pharmacology package that had supported prior approvals. You heard Doug say one of his clients was able to reference the conclusions, in the labeling for the products that were serving as the reference product for his development program. And in this case, because the previously marketed products were for completely different uses, but also being dosed totally different than what we were proposing, we really had to dig in and understand what was applicable. And because it didn't always end up in the approved labeling, where the FDA was really concluding safety, especially for oral dosing, we actually had to contact the original developers of the DFMO products on the market and negotiate right of reference to the underlying data and actually access the reports. In this case, the non-clinical package that supported Ornadil's approval did include a significant amount of oral dosing studies. And so we really needed to be able to access those reports, as well as the National Cancer Institute had done some reproductive toxicology work, which was federally funded and should be made available to anybody that's trying to further public health goals. So we were able to get in contact with them and actually access those source reports as well, negotiating right of reference to include those reports directly in our application. So that's one example of how important it is to really know what it is you need and work the channels to go and actually get the data. The other area we were able to leverage from the historical data was the clinical safety experience with Ornadil at high doses and in pediatric patients. So in this case, we are depending on FDA's previous conclusions of safety and efficacy, since it was broadly indicated at a dose level that will be considerably higher than what we're proposing in pediatric oncology patients. And and so that's very helpful supportive information. And then the last piece, and it's really one of the biggest pieces, is that this product came to our attention by the work that had already been conducted by a nonprofit research consortium, Beat Childhood Cancer, which represents over 40 academic and hospital institutions involved in pediatric oncology research. And through their early work, they had already established pharmacokinetic profiles in patients at the proposed dose range. And they had also already established proof of concept in a phase 1 study, as well as demonstrated preliminary efficacy in a phase 2 open label study. They, through this work, they had already started this work and additional clinical trials that are actually still enrolling today. They also had generated quite a large safety database. So this information, the fact that this information was out there was known because you can research clinicaltrials.gov and you can see that these studies are ongoing. You can see what the target enrollment is. But as Jayanthi noted, the data is not publicly accessible or available. So even though we could see that there was a lot of promise in this molecule, we needed access to the data to effectively create a shortcut for ourselves. So we did ultimately license the data from the research consortium, and we continue in that partnership today. So it is a collaborative development project where industry and this philanthropic organization, as well as the investigators across a number of academic institutions, are all working together to move this to the NDA stage. I can move to the next slide. So the next question, once you understand what you already have and can leverage from the historical work on the product, is what are the remaining gaps and can you successfully address them? Oops.
Sorry about that, a little technical.
No worries. So, in our case, the first gap was there was already a formulation being used in the clinical studies being conducted by the Beat Childhood Cancer Consortium. But to really move it to a registration level, there was a number of gaps that needed to be addressed. So they had really sort of done, I would say, quick and dirty, good enough to get the IND in place and kind of pass the phase 2 mark, but the bar gets raised considerably on CMC when you move to phase 3 and commercial planning. So there was work to be done there. In the area of clinical pharmacology, they had generated some population PK data, which is really helpful, but that data really needed to be examined. We needed to understand PK-PD interactions. There were some gaps to fill, for example, in the area of food effect studies, given this is an oral dosing product and bioavailability, of course, is extremely important. And so we needed to confirm, that we had the right information to be able to appropriately inform how to administer the dose and ensure good bioavailability. And then I would say the biggest gap was that all of the research done to date, it was open label, single arm studies. And the FDA does require that you can support your efficacy claims with an adequate and well-controlled study. There are different ways to support that your efficacy program is adequate and well-controlled. And we determined that we would actually pursue a relatively non-standard route to establishing efficacy, which is to take the existing and completed single-arm study and propose to compare it to an external control database that represents the current standard of care. Luckily for us, in the area of oncology, a lot of the work is, especially in pediatric oncology, a lot of that work is federally funded. So NCI had partnered with another large group called Children's Oncology Group and funded the the database that I'm referring to, or the study that led to the database that I'm referring to as an external control. So we were able to, again, establish an agreement with them to actually get access to that raw data, and thus propose to the FDA to do really robust comparisons of the outcomes in the DFMO clinical trial to this external control. We can move to the next slide. So the other question is, you know, it's nice to dream and you have to dream big, but you really need to think, you know, based on what you can see in the historical information and what you know to be the case about, you know, the disease, can what you're proposing to do really make a difference? And this will also translate to the, you know, the commercial side of the equation, because there will not be adoption and there will not be the ability to get it reimbursed, and there will not be sufficient, you know, market return if it really can't make a difference. Of course, in early drug development, you have to take a gamble or else nobody would ever get anywhere. But luckily for us, we were at a, you know, this program was already at a pretty late stage, considering they had a relatively large study of 140 patients with long-term outcomes available. So what I'm showing here is, and this is, you know, in the literature, but this shows this left panel, and I'll show the right in a second, is the current standard, is the outlook for patients with the current standard of care. So this is a study that looks at the final stage of upfront therapy for high-risk neuroblastoma patients. The dotted line is where that therapy ends today. And you know, it's important to note that when patients achieve remission and they've gone through about 18 months of very intense multimodal highly toxic therapy, and they get to the other end successfully and are determined to be in remission, they receive no further treatment at all. Although it is well established that relapse rates are extremely high, there's nothing currently available to these patients that is well tolerated and, you know, has a good benefit risk kind of equation behind it to continue administering it to patients. But the very sad reality is that that more than 30% relapse in 5 years, and sorry, that might be a little bit off. It really should be probably about 25% die in 5 years. And I mentioned statistics earlier that 50% of patients with high-risk neuroblastoma die in, I think the stats are usually around 5 years. So it's important to note that what I'm showing you here, these are the patients that get through the upfront therapy. So there is a significant proportion of patients that don't make it through the upfront therapy at all. And so these are the best players that actually make it all the way through treatment and are in remission and then still have these very poor outcomes. So if we can show the next panel. So when we look at the phase 2 data, sorry, I'll have to move this around. See what I'm doing? There we go. So when we look at the phase 2 data from the DFMO study conducted in high-risk neuroblastoma patients, and these are patients that had completed the upfront therapy highlighted on the left-hand side and then went directly on to product for 2 years, you can see there is a really meaningful increase, in particular, increase in survival. In particular, you know, I highlight the bottom panel where about 25% of children were dying in 5 years after completion of standard upfront therapy. We have less than 10% that die while they're, you know, as a result of taking DFMO kind of as a preventative maintenance treatment for 2 years after the completion of upfront therapy. And so, and we all know that there's rules, you really can't do cross-study comparisons like this, but this was the look at the data that we were able to you know, have before we decided to move forward with the program. And we determined that that there was more work to do more sophisticated comparisons of this data, but that, you know, at least on the surface, it seemed extremely promising and could make a real difference for patients. We can go to the next slide. So another really big question is, is it commercially viable? So in the case of this program, we asked ourselves a number of questions to make a determination of commercial viability. The first is, do we have freedom to operate? So we did a global patent review and did not come up with any barriers. So this is a very old molecule, which has its pros and its cons. The pro was there's nothing in our way to go ahead with the program. The con is it's very unlikely we're going to develop new IP that's specific to the molecule. And in fact, there was so much interest already in the area of oncology. There's a lot of prior art out in the public domain that basically crowded the landscape as far as even method of use patents. So, but we did have freedom to operate, so we knew we could move forward. The other consideration here is that this program already had an orphan designation, and then when we got involved, it also now has breakthrough designation. But the orphan designation is really attractive because even for an old molecule, going through a B2 route, you can get 7 years of exclusivity in the US and 10 in Europe, as opposed to the 3, for a non-orphan designation following the B2 route, as you heard from Doug. So 7 years we felt was an adequate runway on the market. And then the other questions, which, I do want to just encourage everybody to really think about these because they can really make a difference in commercial viability. So you can approve something for a new indication, but if the product is already out there on the market in the same formulation or same mode of administration, it can be really difficult to get any traction commercially, even though the other product wouldn't have the indication. So, but in this case, there is still no oral formulation in any market for DFMO. And again, since we're thinking about outpatient dosing, the currently available formulations are not a fit. The topical cream, of course, I think it has almost no systemic exposure at all. And then the IV formulation is not, feasible for chronic administration in an outpatient setting. So we knew there were no DFMO products that could be used off-label after we made the investment to get our product approved for use in high-risk neuroblastoma. The other thing to consider is what products are already on the market in the same drug class. So I can share, we have a very specific experience at US WorldMeds where we developed a new chemical entity in a drug class. This is not even a repurposing project, but a true new chemical entity. But there was a generic already marketed in the US in the same drug class, which was prior to the introduction of our product, even though it didn't have an indication, was being used routinely by physicians. So the problem is, that even though it's a different molecule, if there's enough clinical experience, if there's enough literature behind it, and people are comfortable using it for off-label purposes, it's still going to create a really big barrier to adoption because the price point is likely to be a lot higher for a new product coming in than it would be for something that's already genericized. And in particular, it's going to create a lot of payer barriers because payers will not, unless there is a dramatic increase in benefit that they can monetize, they, are unlikely to be receptive to paying for it. And then the other consideration is, what does the market potential look like in dollars? And here, we're really talking about an ultra orphan category with 400 patients a year with the diagnosis. However, we feel like with the data that we have and will ultimately support an approval and be in the label, that we're going to be able to establish a very meaningful increase in survival. And other products in this sort of ultra orphan category have quite attractive commercial pricing. So you're talking about potentially in the hundreds of thousands of dollars for a therapy each year in this kind of a category. So this was our assessment of the commercial viability, and it sort of passed all of the checkpoints. Next slide. So The last thing I really want to kind of ask this group to think about is, what will drive you? I've heard from both of the panelists before me that even though a B2 route technically is supposed to create advantages, and it does, it helps shortcut, I would say, the first part, of drug development, but it is still really hard. A lot of programs fail, and you really need to get passionate. So, I loved what Jayanthi said about working with ALS patients and really putting human context around drug development. And in my case, or for this particular example, my driver was very personal. So this is my nephew, Finn. He is my sister's second child. And he was diagnosed with high-risk neuroblastoma at 3 months of age. And he, so you can see him here on the left-hand side of the screen. And this is, this is what treatment looks like. And this is what treatment looks like during relapse or worse. So thinking that there may be a product that, I can help push that can stop this from being his life in the future, or the lives of other kids in the future that have to go through this for the first time and then get to remission, that is really powerful. So if you can think about the patient community and really just try to, just spend a little bit of time like reading a blog and trying to understand what these patients go through, it'll give you so much energy to push these programs and to, and there's going to be multiple times that you feel like, it's not going to work, but then you have to get the energy to keep pushing, and finding the next thing. So if we can click, at least for now, and hopefully ongoing, we're currently in a very happy place with Finn. He is in remission, and he is actually on DFMO through an expanded use program right now. He is traveling to Charlotte to the study site as we speak, getting his 9-month scans. So he's almost to the halfway point of, his highest risk period for relapse. And so we're keeping our fingers crossed and hoping that our data is, as real as it seems to be, promising. And so that's the end of my presentation. I hope that breathes a little bit of life into what drug repurposing projects can do, and the kind of questions that you can ask yourself as you navigate the process. Just to close out, I'll say we are at the NDA stage for this program. FDA has been receptive to our external control approach to establishing efficacy. And so we hope that this will be on the market next year.
All right, thanks, Kristen, for that, especially bringing up the patient connection, and it's a great story there. A couple other things to mention: the value of collaboration, the value of data, and the fact that it can be licensed. Also thinking about targeting rare diseases. I encourage that thought process as well. So I know that we are kind of over time. I realize that, and what we're going to do is continue this. And for those of you, hopefully, you guys can stay in the audience. Otherwise, if you have something to get to, you're welcome to leave as well. But we'll continue with this and go through a Q&A with some questions. And so I'll go to our next speaker here. Hopefully all our speakers can stay a little longer. I'll go to Bruce Bloom, and Bruce is the Chief Collaboration Officer of Helix Limited. It's a biotech firm in the United Kingdom. They use artificial intelligence, machine learning to look for approved drugs that create novel therapies, again, specifically in rare disease patients. Bruce Bloom is the founder and former CEO of Cures Within Reach, which is a global nonprofit that has brought more than a dozen repurposed therapies to patients via proof of concept clinical trials in drug repurposing. So Bruce, it's yours.
Thanks, Raja, and thanks to the other panelists for their great presentations. It's so interesting. I have been involved with Spark, so a lot of the things that Jayanthi was talking about. I have reviewed the probenecid thing that Doug was talking about. And I worked with Janine Scholler years ago on DFMO. So it's a small community in the world of drug repurposing. If you flip to the next slide, I want to talk about sort of a different way of thinking about and highlight some of the things that some of the other panelists have talked about. And I think about all of the academic researchers and clinicians that I've worked with over the last 20 years in drug repurposing. It's probably 400 or 500 of them. And it's always interesting to figure out how the repurposing got started. Was this something that started as all the way back in some kind of an early scientific research in the lab? Is it an idea that somebody found in the literature? Did it come from a clinical observation? So it's always interesting to think about how this gets started and how that plays into some of the details that the other speakers talked about as far as patenting and other drugs in the marketplace and those kinds of things. So it always is, it's interesting to me, and it often leads back into some of the issues around commercialization versus other kinds of things. It's also interesting to me to think about the different qualifications that the PIs have that are moving academic repurposing forward. Master's degree and PhD, doctors that are doing a lot of the basic science, but reach a point where when things need to start being planned for or move to the clinic, they don't have that experience. Sometimes there are MDs who have the clinical observations and are interested in validating those, but they don't have the basic science capabilities or laboratory space to do that. And then every once in a while, we find an MD, PhD. My experience is those have the greatest success because they can do both sides of that equation. Early on in this, I almost always talk to the PI about what's your motivation? Like, where do you want to see this go? Are you interested in just creating good knowledge for the literature and other scientists? Are you interested in this reaching patients as quickly as possible without worrying about any kind of commercialization? Do you want to start a company? Do you want to, potentially get rich? All of those things are great. I don't have any judgment on any of those, but it's really important. I have been involved in all of those kinds of processes, including times where somebody who had never been the CEO of a company before decided that was something that he wanted to do. And in some of those cases, it's worked out great. In other cases, it has been an interesting experience, but it hasn't been great for moving anything forward. So finding out early what the PI's motivation, and sometimes there's more than one PI, what their motivations are, is really important. It's also critical, I think, to look at when you're doing basic science, how do the models that you're working with recapitulate the human disease? I know that everybody that's on this call understands the frustration with the number of mice that we've cured of all sorts of diseases where it hasn't translated into the human. There's also been experiences in the opposite direction where things failed in the mouse studies or other animal studies that eventually were successful in humans, where we can go back in the literature and look at really good basic science studies where we didn't hit the endpoints, and then people out in the clinical community had patients with comorbidities where a drug that was tested early in the mouse model and didn't work actually helped humans. So I'm not sure that we always get the best answers from the models, but it's really important to understand how well they replicate the phenotype of the human. Clinical observations always helpful, and a lot of the work we did at Cures Within Reach was to try and generate some of these clinical observations as early as possible. Some of the other speakers have talked about, when you think about this as an academic, think about commercialization, so prevalence. How many patients are there out there with this disease? How homogeneous or heterogeneous is the disease? We have a lot of rare diseases that when you actually look at different phenotypes and different subtypes, they really, really become rare. And it's hard to think of a commercial avenue to get something out there when you're carving up a rare disease patient population into ultra rare. There are some other issues between common and rare that we're experiencing at Helix, which is that if there's both a common and a rare indication, in order to make money in the rare indication, you have to charge a certain amount. But if somebody comes behind you with a common indication and charges a lower amount, then how do you separate those price points? And so we think about that from a prevalence and common versus rare indication. Same thing with acute versus chronic. There are a lot of repurposing opportunities out there where it's an acute disease, and it's, you know, unless the incidence is really high, it's hard to make a lot of money in the acute space. A lot of people, you know, say that the pharma industry is actually not there to solve health issues, but to just help them endure. You know, there's so many medications out there that are much more chronic and keep people in a chronic state. And so we think about those kinds of things. A lot of the other speakers have already mentioned the off-label versus commercial use, so I'm not going to talk about that or some of the pricing. One other thing that I think is really important if you want to commercialize is your ability and your attitude about working with industry. So, you know, making sure we're on the same page about who's going to do what, who maintains control, how much money's going to pass in one direction or the other, how much money gets paid up front. All those things are important considerations when you're thinking about this. And, you know, Raja's job and the other commercialization officers and people in the tech transfer department is to help guide those kinds of discussions so that the parties on both sides are, you know, are really aware of the kinds of sort of standard agreements that happen in those kinds of situations. And then the last thing that's important is, can the patients participate in studies, especially in a lot of rare diseases, but in some common diseases, patients are so sick or it's so difficult for them to travel that, you know, participating in the studies gets really challenging. Also, if you've been in one study, you're often excluded from other studies. And again, in the rare patient population, there's often a real challenge there about getting enough people to participate. So we go to the next slide. I just want to run quickly through some examples of different kinds of repurposing participants and what outcomes, and just sort of contrast and compare the differences. So we've been involved in a lot between just philanthropy and academia. So if you click, you'll see one is this sirolimus for acute lymphoproliferative syndrome, which is autoimmune lymphoproliferative syndrome, which is an autoimmune cytopenia that kids have. This was a project that we funded at Cures Within Reach. So within 3 years, we funded first to build a mouse model, show that sirolimus worked in the mice, and then did a proof of concept clinical trial. So in less than 3 years and in under $300,000 for all the total work, we published results, and now this drug is used off purpose for ALPS all around the world, saving lives and a considerable amount of money. And it turned out that we did a second round of research in 5 other autoimmune cytopenias, and in 4 of them got the same results. A relatively low dose of sirolimus taken twice a day really helps these kids. So this was fast and cheap. 3 years, under $300,000. And if you click again, you'll see another repurposing. So this is for the vaccine BCG being repurposed for type 1 diabetes. This is with Denise Faustman at Mass General. This is now probably in its, well, we funded the first time in 2006. So that was 15 years ago. And the total cost so far of the now phase 3 trial that she's doing to get regulatory approval, it's over $25 million. So these are 2 seemingly similar philanthropic support of academia, not trying to get any kind of commercialization, but they look totally different. And it's partly the patient population. It's partly what the researcher wanted to do, but two similar but totally different outcomes. If you click on the next one, you'll see philanthropy, academia, and government. So flipping again, you'll see there's two different things I want to compare here. The first one is Oranafin, which is a medicine that's made out of gold, repurposed for chronic lymphocytic leukemia. And this was between the University of Kansas, the Leukemia and Lymphoma Society, and the NIH, all worked together to do this repurposing of Oranafin. And there was no real commercial viability for this. And this was a project that took about 5 years to get moving, and then another several years to get it together. And at a certain point, it just completely stopped because there were better treatments out there for CLL. The other one, metformin, being repurposed for TB, started with a group in Singapore that did a retrospective study that showed that type 2 diabetics that were taking metformin had less incidence of TB. If they did get TB, they had a shorter course before cure, and many less went into becoming in the resistant category. And so now a nonprofit called the Open Source Pharma Foundation, which I work with, and the Indian government and the Tata Trust actually are performing a prospective study adding metformin to the 4 drugs that are currently used for TB. If you click again, you'll see that there's patient groups, academia, and pharma. So clicking again, this is a repurposing of nitisinone, which was originally a pesticide, but turned into a drug, repurposed for the drug, the disease AKU, which is also called the black bone disease, alkaptonuria. And this was, again, fairly long, 15 years from start to finish, cost about almost $20 million to do a phase 1, phase 2, then a second phase 2, and a phase 3 trial. And last October, it got approved. And it's really a wonderful therapy for the kids with AKU. And then biotechs in academia. So I helped start an organization called Rediscovery Life Sciences, which is a biotech. They're repurposing the drug Suramin, which is an African sleeping sickness drug for acute kidney injury. This was research that started at South Carolina, University of South Carolina. And so that's moving along. It's in phase 2 clinical trial. And then if Rediscovery Life Sciences gets the right results, they'll then move that to a pharmaceutical company. And then one more click, and we're sort of towards the end. So Raj, I'll turn it back to you, and we can start with questions.
Yeah, thanks, Sarah, Bruce. Thanks for that presentation and all the different examples. So yeah, we can move into the Q&A and go ahead with those questions. So one thing I wanted to mention is that to all in the audience, that you could see that we've presented with all the speakers the challenges that are there, but that's not to dissuade anybody from advancing these technologies, but is more to again keeping the end in mind and thinking about all the different possibilities or different obstacles that could come in the way, so that as researchers you could think about these things again way in advance. So bringing all those things up front is the reason to talk about these and talk about all the different challenges. One of the things that I think Bruce hinted at it in the beginning, and I want to touch on, because this idea of off-label, and I think there might be researchers there that might just kind of advance existing commercially available FDA-approved products that at the given dosage, through for repurpose indication all the way through clinical trials. and they may go through that process and just think, yeah, there's a lot of benefit in doing that. But I wanted to touch upon what could be the disadvantage. I want everyone to kind of realize what might be the disadvantages, of doing that work, and will it, are there instances where it may not, the value may not be as much, and essentially, what are the disadvantages of off-label? I don't know, Bruce, you want to start with that one?
Sure, I can speak to that. Yeah, well, certainly the advantage is that if you can publish some clinical results that show that something works off-label and there's an unmet medical need and the drug is available and inexpensive, it often, at least it's available out there for physicians and patients to decide if they want to use it. And in the rare space, it often gets used fairly often because there's nothing else to treat the patients with. On the downside, you certainly don't get the same kind of penetration in the physician market with an off-label use that you get with an on-label use. There are physicians who won't do that because there's professional liability risk. There's physicians that won't do that because they're not convinced of the safety. There are hospital networks and others that won't put it on the, won't let the pharmacy distribute it for that kind of thing. And then maybe the biggest hurdle, especially if it's an expensive drug being used off-label, is that it won't get paid for by, at least in the US, by the health insurers. And many of the ones that we were able to move to patients at CureSwift and Reach, what we found was the patients ended up paying out of pocket at the beginning to prove that this had efficacy and improved the patient outcome and reduced the healthcare costs. And once the healthcare company, the insurer, saw that, then they were open to pay for it, because in many of these situations, it reduces healthcare costs significantly when the patients go on these repurposed therapies. But those are just a few of the things that make it challenging for off-label use to be successful.
Okay, great. Sounds good. I know we're going a little over here, and I'll just maybe throw in one or two more questions really quick. One maybe for Doug. I know you've seen a lot of different ones, and you talked about in your case studies. In your experience, do a lot of the repurposed drug assets that are moving through approval typically require those bridging studies, or what are your thoughts about that, and in terms of researchers and what they should be prepared for?
Yeah, certainly if you want to rely on FDA's previous finding of safety or efficacy, you need to bring forward a valid scientific argument of why your new way of new indication or new way of delivering the drug is similar enough to the old body of data that's out there on the previously approved drug that you can rely on some of that information. And so that's called bridging in the regulatory sense. How does your asset align with the old one so that you can rely on some of that data? And mostly, if you're looking at a new efficacy, you'll want to rely on the previous finding of safety. So you'll show that your method of delivering it is very similar, blood levels, exposure to the previous method. You'll do some animal studies to perhaps confirm that, but that's the bridge. Sometimes you may want to bridge to efficacy if you are delivering it a new way, but you can match it's bioequivalent to the old one, and then you can bridge to the efficacy data and then rely on the efficacy data, so you don't have to complete full phase 3 clinical trials to prove efficacy.
Okay, so I'm going to go with one more and then a comment, but this one is going to AI and ML, and they are aiding obviously in advancing novel drug discovery and drug repurposing. But I want to know what might be some of the challenges to that, and are there possibilities that maybe different AI platforms can give different answers, let's say. But maybe I'll go with this one a little bit from Bruce and then also from Jayanthi, just a couple comments there.
Sure. AI, I think, is great. It isn't proven yet. There isn't an AI predicted drug that's made it across the finish line yet to patients, although there's many out there that are in the preclinical and clinical trial space. Absolutely, you're going to get different answers from different AI depending on what the algorithms are, what data is in there. The biggest challenge right now, I would say, is that, two big challenges. One is it's hard to get all the data in the right format, even when it's available. And the second is that there are a lot of data that are not yet available. They're not published. If we all had access to all the data, we'd make much better predictions. And I guess I would say maybe a third one that I'm thinking about as I'm talking is a lot of AI use published literature and published data. And if you think about the number of research projects that have come up lately where the second or third try hasn't validated the research result from the first try, you wonder how accurate the data are that we're all using to make our predictions. And having said all that, the speed and breadth and depth of AI being able to do stuff that scientists take a long time to do, and even some of it where scientists just wouldn't have the ability to connect all the dots, I think is really important. And at Helix, we use that to connect all the dots, and then we show it to really smart scientists to say, do all the dot connections make sense? And so it's still part AI and part HI, human intelligence. We haven't got to just solely, AI and ML. Jayanthi, what do you think?
Sure. I think Bruce has covered it very well. I think I can say that it is AI ML is excellent, but it's still predictive. So it's still going to need all the experimental evidence that we need to generate to have FDA approve any of the compounds. Having said that, there are several companies out there. The only problem I feel is that most of them are using customized tools. So there is the data that's coming out is not uniform for a common academia to interpret and access. Accessibility is still very limited. They are not shared. But what the good points I feel is that through AI, you can handle large amounts of data sets out there. Literature and text mining becomes a lot more simpler. You can also apply a systems biology approach where you can connect several different databases, the pathway analysis, the genomic, all the omics data that is coming out. It's very, very easy to apply all of them together with AI and ML and have something come out useful for patients. It's very educational, and it can certainly minimize cost because of its excellent application in early stage drug discovery. In the medchem part, I think it has a lot of value.
Okay.
It's a great idea.
Yeah, so I appreciate all the speakers' times and their presentations, very insightful. And also all the audience for attending. And this is the culmination of the 5-part series, and I found it very exciting, and I learned a lot myself. So again, thank you, everyone, and I look forward to it next time. Bye.
Bye. Thanks, everyone.
Good afternoon.